here is going to be fasciculata. And then if we keep moving down, so medulla is right here. Again, you can see the boundary. That's the edge of the medulla. Then the layer of cells right beside the medulla would be reticularis. Down here's the capsule again. And that layer of cells right by the capsule is glomerulosa. Right here. And then all of these cells here in the middle, that's fasciculata.
What's the area that's kind of jutting inward, like that weird pink thing?
This right here?
Yeah.
Is probably just the tissue probably folded when they were staining it, so it looks a little bit darker. But you should be able to kind of see that glomerulosa looks a little bit darker, and that's because the cells are kind of clumped together a little bit because they're kind of oriented like this. So they're kind of close together. Fasciculata, the cells are more like this. And then reticularis, they kind of go out a little bit like this, and there's more space in them. So I'll either show you this at 4x or 10x. If I were to show you at 4x, I would definitely make sure that you could either see the capsule or you could see the medulla so that you can always reference it. You see the capsule, cells right beside it are glomerulosa. You see the medulla, cells right beside it are reticularis. Anything in the middle, fasciculata. Okay. Questions on endocrine?
Can you go up to 10x on that?
Yep. So 10x, let me go to the top here.
It seems like it would be harder to see the layers.
It's going to be hard projecting it like this. So this is at the top again. So this is the capsule. And again, you can see the fibers running horizontally. And then here you can kind of see swirl, 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 swirl. That's all glomerulosa right here. Glomerulosa. Down here at the bottom, the cells are not, here they're kind of swirling a little bit, but down here they're going to stop kind of doing this swirling orientation, fasciculata. This is all fasciculata in here. Down here, I know we're getting into reticularis because the cells are kind of going at angles a little bit. And you see more white spaces in here. So I know we're getting into reticularis. So the cutoff is probably right around there somewhere. Kind of right in there. So fasciculata, reticularis. If we keep going. This is the medulla down here. There. So this magnification projecting, it's a little bit hard to see because it's hard to see point of reference unless you're way at the top or way in the middle. But when you look into it, you can see more. This is actually, this is a good dividing point right here between medulla and reticularis. That's a good dividing point. And I'll always try to show you, like here, this is a better dividing point between medulla and reticularis than up there. So I would, if I wanted you to say reticularis, I would show you this area where you can really see the distinction between the two. Again, if we just keep going down. Reticularis is at the top, and then we're going into fasciculata. And then down here again, the cells are starting to look a little bit darker because they're kind of clumped closer together. They're kind of swirling around. That's glomerulosa, and then capsule down here again. Switching over to the male reproductive system, I'm going to do the spermatic cord first. Spermatic cord is going to be easier to see at 4x. Maybe 10x, but 4x is usually the best. So when you scan around here at 4x, again, you're looking for arteries, veins, and the ductus deferens. So when you scan around here, you want to look for the biggest circle possible, and obviously you guys won't be scanning on the test. It'll be at one spot. But when you're studying, you want to look around. That is the biggest circle in that slide. This is your ductus deferens. So it's going from here over to here. This is the lumen right there. Get that focused a little better. So by far, that's going to be the biggest circle that you see in that slide. And the other way I know that's the ductus deferens is it's got this really huge layer of smooth muscle cells. So if I would magnify that, I would be able to see that these are smooth muscle cells going this direction. And remember, smooth muscle cells are not striated. They're tapered, so they'd be fatter in the middle and taper out to the ends. And the nuclei would be right in the middle of the cell. I can also see that the lumen here is lined with epithelia. These are the cells that look darker purple, and they look darker because they're so close together because they're epithelia cells. And you can see that these have kind of a columnar shape to them, particularly when you go to compare it to an artery or a vein that would be next to this. What you're seeing in here, these are actually sperm cell here in the middle. So you may have a ductus deferens with sperm cell in it, you may not. Just depends on that particular organism, what was going on with the sperm cells at that particular point in time. So that one you can see, huge layer of smooth muscle, very circular, lined with simple columnar epithelia or pseudostratified, one of the two. See some sperm cell in there, but again, you don't necessarily have to see sperm cells. If we move over and then look at arteries versus veins, this is a good section here. This right here would be an artery. So again, you can see it's very round, but it's not nearly as big as what we just looked at. Here, you've got a layer of smooth muscle, but not nearly as big as what we just looked at. You've got epithelial cells lining the lumen, and these, you can see, are much shorter than the ones we just looked at. So those would be simple squamous epithelial cells. And then all around it are a bunch of veins. This would be a vein right here. This is part of a vein. Here's a vein right here. Here's a great big vein right there. This would be vein, that's probably a vein also. So again, when you look at veins, veins are going to have a bigger lumen than the arteries. They're also lined with simple squamous epithelia. The layer of smooth muscle is going to be much smaller than the arteries will be. So again, take a look at that, and then we'll go back over to the ductus deferens to compare it. That's the ductus deferens.
Do you want us to remember simple squamous epithelium for the veins?
No, I probably won't ask you that, but probably won't ask you that, no. It'll come up again when we do digestion, so probably should learn it at some point, but I won't specifically ask you. What you're looking at right here, this is also an artery, and this is also a vein. Why these look a little weird is there's dried up blood in the middle of them, and that's giving this kind of pink appearance in here. So those are just blood cells that are still, that just got stuck in the vessel. But again, this is an artery because it's very round, layer, smooth muscle. This is a vein because it's more oblong, layer, smooth muscle, smaller, lumen's bigger in the vein than in the artery. This would be another vein right here. This would be a small vein. This would be another vein, little artery, and your nerves are going to be these guys right here. They're going to be smaller circles that are just solid, and they don't have a lumen in them.
Can you see the microvilli? Or
The what?
The microvilli, or do you have to?
No microvilli on these guys.
Am I reading the wrong thing?
Yeah, no microvilli on these guys. They're just transporters,
Okay.
right? They're either transporting blood or they're transporting sperm or semen. So no microvilli on these. Okay. Then if we go over to, let's go to the epididymis. These are the guys that you'll see microvilli. Really long microvilli, and they're actually called stereocilia. So this is at 4x, and so this is a cross-section of the epididymis. So you see tubule, 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 tubule. So all of those kind of circular areas are the tubes that the sperm is swimming through, right? And so you cross-section it, and so you just see all these little circles. And in a lot of these, you see sperm, so that's what all of this here is in the middle. Those are all sperm. And generally, you're going to find more sperm in the epididymis than you would the testes, because sperm go to the epididymis to mature and hang out and wait. Chances are you will see some sperm in the testes as well, but you're going to see just sperm that are releasing, traveling through the testes to get here. Now, when you look, we need to magnify this to really see it, but what we're going to do is we're going to look here at the wall of these tubules, and the wall of the tubes of the epididymis are going to be smaller, not as thick as they are in the testes. And again, that should kind of make sense, because remember, in the seminiferous tubules of the testes, in the wall, is where we're doing all those rounds of cell division. So you would expect the wall to be a little bit thicker. And when you look in the testes and you look in the wall, if you look, magnify it and look at the nuclei, you should even be able to see the chromosomes actually condensing. They'll look like little strings in the nuclei. And that's because the chromosomes are undergoing division, right? They're going through mitosis or meiosis. Now, when we magnify this, you should see that the edge of these cells here are not going to be clean at all. And that's because there's lots of these long microvilli, and they just make the inside of the cell look really, or the inside of the tubule look really torn and ratty. So I'm just going to magnify this up to 10x. Just find a good spot here. And again, it's a little bit hard to see projecting it like this. You can see the microvilli better if you just take the fine focus and go forward just a little bit and back just a little bit. But you can see these kind of, it looks kind of ratty, or these things kind of sticking up. That's the microvilli here of the cells lining these tubules. So all of those little frayed areas, those are the microvilli. And again, what's inside of here, those are the sperm cells in there. So again, you can see that's a long microvilli. There's some microvilli. Now, if I increase it up to 40, again, you can see. So you can see here the wall is not real thick. You can see this, these are the microvilli coming off here. So for the testes and the epididymis, it's better to show it at like 10x or 40x, because then you can really see these cells that are lining the lumen. So if I switch that and go over to the testes now, here you can see that the wall of these tubules is much thicker. So again, remember in the testes, they're very slender but tightly coiled seminiferous tubules. And so that's what you're seeing here. Here's a tubule, here's a tubule, here's a tubule, 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 so on and so forth. And the wall here is very thick because those nurse cells are here in the wall of the seminiferous tubule. And all those rounds of cell division, this would be the basal surface, and those cells, the primary spermatocytes, secondary spermatocytes, spermatids would keep moving closer and closer to the lumen down here. So if I magnify this up to 10x, find a good one to look at here. So I'm looking at this one right here. So you can see that the inner edge of those cells is much cleaner than what we were just looking at. And that's because these guys don't have microvilli on them. You can also see that you can see the nucleus real distinct in these cells here. And when we go up to 40x, you should be able to see that in some cases you can see the chromosomes moving apart like this. In some of the nuclei, you can see the chromosomes lining up in a line. And those are all those different stages: prophase, metaphase, anaphase, telophase. And they'll look like little strings. Instead of just being a purple nucleus, you'll be able to see like little strings, the little chromosomes actually inside of there. And again, you can see that because that's what's taking place here, spermatogenesis. Now, if we magnify that one more, go up to 40x. So again, when you look in the nucleus here, all those little strings, those are the chromosomes. You can see them all in there. Those are all the chromosomes. And then what you're seeing here, these are the sperm detaching from the wall and going in. So these are the tails of the sperm that are detaching. Here you can see the head of a sperm right here, and then the tail is right here, and it's getting ready to detach. So, again, really spend a little bit of time between looking at this versus the epididymis so that you can tell the difference between a really kind of ratty look to the inner layer of cells as opposed to tails hanging off of the edge of this area lining the lumen. But again, what I look for is this really thick layer, and you can see these cells very distinctly, and you can see the chromosomes in each of these cells. You can see it real clear. And again, the edge of the cells looks much cleaner than the epididymis does. Questions on the male system? What did I miss? 9:02. Okay, we have a couple minutes. So let's go over to the ovary. What you want to look for in the ovary is you want to look for different size follicles with oocytes in those follicles. And the follicles are just groups of cells that house the oocyte. And they're going to be on the edge of the ovary. So this would be a small follicle. This is a really big follicle. That's a pretty good sized follicle. That's the oocyte inside of that follicle. Let's keep going down here. Here's another. So this whole thing here is the follicle, and then that round part in the middle, that's the oocyte. So during ovulation, what's going to happen is the follicle is going to rupture, and that oocyte's going to come out of the follicle, out of the ovary, and then get swept up into the fallopian tubes. So you'll see different sizes because they're in different stages of development. And I'm not going to ask you to know, is that a primordial follicle, a primary follicle, a primary oocyte, a secondary oocyte, but you should be able to see, again, on the edge is where you're going to see these follicles, and then the oocytes will be in the middle of them. So again, just always look out on the edge. This is the best one on this one right here. But these guys are small follicles, and that little circle in the middle is just a tiny little oocyte that's early in development. This is getting close to the point where it can be ovulated. So that's the ovary. And again, that's going to be best to see at 4x or 10x, so you can really look at the outer edge of the ovary. So then if I switch over, look at the vagina. The vagina is going to be easy to tell because it's going to be the only structure that has stratified squamous epithelium. And so when you look here, this would be the innermost part of the vagina right here. You're looking at just one half of it. This would be the epithelia that lines the inner part of the vagina. And again, you should be able to see layers of cells stratified. The cells look like they're kind of squashed, squamish. These are really durable cells because the vagina has to deal with a lot of abrasion. When you go from this part down, this is the smooth muscle of the vagina, and then these little circles here, those are just glands that are releasing mucus. So those would just be rings of simple cuboidal epithelia. But the key thing that you want to look for is, for the reproductive system, the only structure that has stratified squamous epithelia, that's the vagina. Now that, you should know stratified squamous because it's a key identifying feature of the vagina. So that I would definitely, if I pointed to that, you should be able to say that that's stratified squamous epithelia. And because it's stratified squamous epithelia, that's the vagina. So that one's going to be the best to see at 4x. If we went up to 10x, 10x is good to see too. So again, you can see the cells real nicely. Many layers, cells look like little fried eggs, little purple nucleus right in the middle, right inside of them. The last one is the uterus. Let's see if we can get that up here before they shut us down. For the uterus will look different depending on what cycle it is in the month. And so this one right here, this would be the innermost part of the uterus. So this layer of cells right here, this is part of the endometrium. And that layer of cells, we're going to talk about this on Thursday, this will either get thicker or thinner depending on what part of the cycle we're in. So during menses or menstruation, this part of this endometrium, except the very deep